Morning, everybody. It's Mike here, and today we're going to take a look at stress and just go back a little bit. Most of you guys are coaches, or perhaps all of you guys and gals are coaches, I suspect. And this all started about 5 years ago in Saskatoon when I was asked to do a little presentation on stress, and some of you may have been there. And that kind of led me to, for the last about 5 years, really gearing up on how stress is handled in plants. And I think I finally understand a big chunk of it. So I want to discuss this with you today. As usual, I have a lot of pictures, so I'll move through fairly quickly. I'm going to do a little bit of physiology, and I was telling Jill that I'm going to put together a special webinar on just basic plant physiology, so I don't have to go through this many times over my my speaking career. Anyhow, I always start out with this one because you guys and gals basically do your agronomy by looking at nutrition and application of nutrients and understanding what's going on in the field. And we have a technology now that way back 40 years ago wasn't available, and that's called the tissue test. When I first went to California doing my MPM out of Simon Fraser, you know, the only people doing tissue tests were potato people and orchard people, and it was amazing that it didn't come into grains. But now with technology, it makes it economical to look at tissue testing along with soil testing. Today's presentation is going to deal with those underlying compounds: oxygen, potassium, sulfur. All the other ones are important, and I always make that statement. It's not that these are the other ones aren't as important, that just for stress management, some of these are more important than others. Iron, manganese, zinc, copper, and nickel. That's quite a few. Many micronutrients, and you'll understand why. And then in the macros, there's secondaries, the sulfur and the potassium. We always need lots of nitrogen and phosphorus, which build the cells. And as usual, we always got to get back to the cell, because the cell is made up of the plant is made up of millions of cells. And unless you understand what's going on in each cell, it makes it very difficult to really make a decision about what you're going to do. And that's why I love the tissue test. And it struck me, you know, as a human being, over 7 years of age, I've had umpteen blood tests. A plant goes through the same life cycle in 1 year. We give it a little soil test to find out what's there, and then we might give it one tissue test to find out if it's doing okay. And if you think of it for any length of time, it doesn't make sense that we don't do more tissue tests, because that's really our signal of what the plant is getting. So let's get on with it. First off, about 80% of our soils are phosphorus deficient. And I was on tour of Saskatchewan last week, and I'm amazed at a lot of central Saskatchewan that we have drained our P levels down to 2 and 3 parts per million in many areas. And it doesn't seem as though the calcium is tying it up as much as we thought, because there's a lot of high calcium soils and high pH soils, and there's no phosphorus in it. So unfortunately, that is key to the whole plant functioning in terms of building the cells, along with nitrogen. And phosphorus and nitrogen really regulate how the roots are going to form and how roots are going to grow in that whole source to sink process. Again, we want to take a look at this, and I'll just use my cursor here to point out some things relative to what I want you to look at. We don't seem to have much problem with nitrogen. Everybody's cognizant of the demand of nitrogen and how much nitrogen we're moving, but let's look at P. And let's look at the amount of P that canola removes. On 21 million acres, we have about a pound of P leaving per bushel. That's an uptake of about 15,000 tons per year on just canola. Never mind what we're removing in wheat, soybeans, or peas. So that's a really big amount that that's leaving our land to build cells and provide the energy. Let's take a look at S. Sulfur. Now let's look at K first. Tremendous amount of K taken up by the plant, almost as much as nitrogen in many cases. Not quite as much, but a lot, but not much leaving the plant. But K is key to stress management. Sulfur, same thing. Look at canola. The amount leaving relative to everything else, and I'll show you something later. Zinc. We're losing zinc, and take a look at things like peas take up a lot more zinc than other plants. Every plant has different capabilities of taking up zinc, for example. But you can see that canola takes up a lot of zinc, wheat takes up zinc, they all take up zinc. Take a look at manganese. Soybeans take up a lot more manganese than any other plants, and that's because of the way the soybean plant handles the movement of its nitrogen from the nodules up into the cell tissues, and it has to convert it from what are termed ureates into urea and then assimilated by the plant. And that process of change takes a lot of manganese, but all of these plants are moving a lot of manganese and taking up a lot of manganese. Not much leaves in canola, but it takes up a lot. Copper. Everybody talks about copper in wheat. This is spring wheat here. And it does take up a fair amount. But canola takes up more per bushel, and yet we don't see deficiencies in canola because canola can suck it up and is a scavenger beyond belief. And lastly, look at iron. If Ryan is listening, I'm not sure I pronounced it right, but it's okay. Look at canola and iron uptake. Huge amounts of iron are taken up compared to peas and other plants. So every plant, the message is, every plant has different capabilities and different needs. And when the plant is under stress, it pulls on many of these nutrients in order to deal with it. And that's what this is largely about. What nutrients and how do the plants deal with it? So this is always a good guide to tell you which plants require which nutrients. And when you look at tissue tests, take a look at the demands of those plants, because that'll help you guide what's important. And we got to have these nutrients all through the life cycle of this plant. And especially here at bloom, when the plant changes from building tissue and building reproductive parts to actually blooming and starting to grain fill, because then we reduce the ability of plants to pull up as much nutrient and send it back to the roots to support root growth. And we need to have that water, because water drives everything, as we all know. And you can see that that peak also diminishes after heading in flowering, because at that point in time, the plant is now starting to senesce and dry down and fill pods. So it's moving a lot of stuff. Now, the key thing is you need water here. You need water to move these nutrients as plants senesce. We grow what are called monocarpic plants. All of our crops are annuals. They reproduce once in a season and die, leaving their seeds. So their strategy is to build this flower, build a seed, and leave the progeny their remnants. The mother plant leaves the progeny, the seed, with a full load of nutrients, so it pulls everything out and puts it into the seed, and the seed carries on to the next year. We use all of these nutrients in this buildup here, and then we sort of maintain them through that, and then we pull them into the seed as time goes on, and we need them right up to the end of life. So we have to have that plant functioning. And my thesis is, as long as we look after photosynthesis and reduction of stress, the plant genetically knows what to do. This just goes to support again that we are moving our phosphorus off. And if you look at, this is wheat, but if you look at soybeans, you look at many of our crops, almost all of them move the phosphorus into the seed, because the N and the P go into the seed, because the mother plant needs to provide that to the young seed and the seedling to start building its proteins and give it the energy to grow. So it makes common sense that virtually all of our monocarpic plants, like wheat, canola, soybeans, all of those plants, oats, barley, all of our plants will do that. So we will continue to lose those nutrients forever and a day, whereas potassium is used more to stimulate things in the plant. So let's get on a bit to stress. You've seen this, we don't worry about that. We know we've done a good job of biotic stress loss. We do a good job of spraying weeds. We do a good job of fungiciding plants and controlling diseases through disease resistance. So we've done a pretty good job, but most of the abiotic stress we have real difficulty with, and that is one of the limiting factors, if not the limiting factor, to us increasing our production of food on the globe. So let's take a look at the plant. How does the plant deal with anything? Well, it deals with it genetically because it can't run away, it can't hide, it can't do much. It can turn its leaf, it can grow some roots, and it does all that genetically, and it can change its chemistry. So well, let's take a look at some plants here. We got, gene numbers, and, we got corn, we got about 32,000 genes. Soybeans got a lot of genes, 46,000 roughly. Arabidopsis is a small plant, about the same genome as corn, not that much. This is an old Brassica rapa. We got 41,000 probably. In our hybrids, we're closer to 50,000 nowadays. So these genes are up and down regulating everything in the plant in response to the environmental stimulus. And some of those stimulants are stresses, heat, drought, those kind of things, which I'll get to in a second. But it's basically done genetically. Just to give you an example, I was at the club root meeting at the university about 2 weeks ago, and one of the new grad students, postdocs, was talking about resistance in canola, and I said, well, did you notice an uptick in the gene regulation of salicylic acid? He said, yes, we did, but it wasn't enough to handle the attack by the clubroot organism. Well, it just so happens that salicylic acid is the primary hormone that comes into play when a plant is going to attempt a systemic response to a disease. So this is how the plant is genetically managing virtually everything in its daily life by up and down regulating genes and making proteins, because all these hormones are proteins, whether it be in germination or in senescence or in disease response or response to grazing. The genes are changing up and down. And if you can well imagine across a landscape, each plant is facing a different environment, both in the soil and in the air. And each plant responds differently based on the signals it receives. And the other thing is that there's always a source sink response. The source are leaves that's producing the energy. And when it's a seed, it's the energy, it's the source for the new seedling. The sinks are the flowers and the roots, because a root can't feed itself, so it's always a sink. So if the sink needs more of something, sugars usually, because that's what photosynthesis does. And then within the cells, there's a biosynthesis system that uses the carbon to make all of its molecules. So if the sink is demanding something, that means the source, the leaves basically, or the seed if it's germinating, have to give it to them. And in order for the two to link, because the source and the sink have to be coordinated, because if the top is growing but the roots aren't and can't supply, the top has to tell the roots, man, I need more something. So the hormones come into play to do that. I'm doing some basic stuff because it's a good time to review, and we're starting out with EDS in our first sort of production webinar. Now, the plant, of course, takes up nutrients, is a liquid in liquid format. It moves up the xylem into the leaf, and I'll show you this in detail. It runs it through photosynthesis, gives out oxygen and sugars. CO2 goes in, the sunlight runs the photosynthesis, and it puts the sugars into the phloem, which move up to the new growing points because they can't photosynthesize, and down to the roots because they can't photosynthesize. And it's controlled by the shoot apical meristem and the root apical meristem, that source-sink relationship. And they're communicating through the system, the xylem and phloem, and intercellularly on those messages. And that's all run by water. It is the water that is transpiring out the leaf and coming up to xylem that brings it the nutrients in terms of minerals and water up into the leaf, where then it can produce sugar. And it's here in the leaf where the problem kind of starts. If we're in the drought stress, cold stress, hail stress, the plant is losing water and it shuts the stomata. Now the stomata allow CO2 in and they allow oxygen out. When this happens and it closes, the oxygen becomes toxic inside the plant, and as it accumulates under stress conditions, it causes damage. The two places that let me go back. The two places that occurs in are in the chloroplasts and in the mitochondria, and I'll cover that in a few seconds. So here we have the root. In terms of the root, it is also facing stress down here. As the tip goes through the ground, it's running into pressures, it's running into disease organisms, it's running into nutrient deficiencies, and something like calcium is picked up almost exclusively right back here, right back at the growing tip, and here right next to where the lateral roots form, where the Casparian strip is broken down inside the root. So we're getting root organisms that are both beneficial and negative, working on all these root hairs, but it's the exudates that the plant is putting into here that are feeding microbes and also putting proteins in the cells in order to extract nutrients and water from the plants. So the root is key. And what do we say? Gerald Anderson, God bless him, says if you want to grow roots, put on phosphorus. And he is so right. So then we take that nutrient when we run it up the root. This is the wheat root, and this is the rhizosphere. This is all it can get the nutrients from. So nutrients that are immobile, this is as far as they can get unless they have some mycorrhizal association. Nutrients that are mobile, like say a nitrogen, a nitrate, can move through the mass flow and can come from great distances. And the plant is always struggling with that. But if it's immobile, the plant has to grow to that nutrient because the root hairs are where most of the extraction of nutrients and water takes place. And then moves it into the xylem and phloem up the plant. You can see wheat here, you got xylem and flowing. This is soybean. And the main stem moves up the laterals, up into the base of the leaf. And you can see this is wheat. This is soybean. And you can see there's always a face looking back at you when you look at these here. And this is the phloem here, and the xylem moving up into the soybean leaf, and it's just full of sugars. Moves up into the veins, the nutrients, into venules right to the tip of the leaf. And then as it photosynthesizes through all this, it moves the sugars back through that system, through the plant. And it's these little stomata where air and water and carbon dioxide enter and leave. And we get stress from things like this. This is disease on the leaf. So what the plant does when it gets this disease, and you can see it's sort of dying off here on the edges, and I'll introduce you to the term, it produces reactive oxygen species, primarily hydrogen peroxide, from the cells in response to the attack. And it puts it into these, around these cells, the disease cells, to try and block it. And the best example I've come up with is this. We're all familiar with hydrogen peroxide, H2O2, when we get a cut. And we got kids, when they get a cut, we put some hydrogen peroxide on it. What do you see? It's fizzling. It's oxidizing both the microbes and the tissue around the wound. And in that oxidization, it kills the microbes. This is the same reaction the plant has in terms of responses. So these reactive oxygen species are normal in the plant in response to stress. Now, at the same time, the plant is always trying to keep a balance between good stuff, bad stuff, things that are high, things that are low. And that's what homeostasis is. Oops. So, and it avoids any toxic compounds by adjusting the level of minerals between the vacuole and what is called the juices within the cell. Its fancy name is cytosol, but it's the juices within the cell. So that's where the plant moves its toxic materials into the vacuole, and that's where it also stores stuff. So here's the cell. I got this from Tim Myrick back a few years ago, and it's the only one I found. I'm not sure where he got it or he made it up, but it's a nice one. And all this shows you is that within the cell, in the mitochondria right here, and in the chloroplast right here, we've got all of these ions, copper, calcium, magnesium, again, zinc, all of these in here working everywhere. So the plant is moving this in and out, and anything that's spare or toxic moves into the vacuole where it's safe. So the key things for stress. Now we're getting down to nitty-gritty. The chloroplast here, and the mitochondria here, these little guys here, mitochondria. I'm going to be talking primarily about oxidative stress, because when you look at the stress in plants, the response seems to be the same at the cellular level, no matter what the stress is. And it occurs because of this. In the chloroplast, the plant has got an electron transport system, and I'll show that in a minute, but it's got an electron transport system that produces ATP and sugar. At the reverse side of it, in the mitochondria, which provide respiration, take some of the sugars produced here at night and during the day when there isn't enough, and it respires because it needs to make energy for the cell to operate. Both of these are where the electron transport system perform, and that system is not 100% efficient, so you get oxygen atoms that are modified and become toxic to the plant if they are not controlled. But at the same time, those are useful to the plant in response to certain stresses. Depends how bad the stress is. So those are the key parts of the stress response and where the stress occurs. This vacuole is where everything is stored, and it'll fill up about 80% to 90% of this whole cytoplasm, and all this liquid is the cytosol that was referred to, and here's the nucleus. So the plant to have homeostasis must have an ability to control what goes in and out of the cell, and it does. This is called the plasma membrane. And the plasma membrane is just this tiny, tiny ridge here. I'll show you a better picture coming right around the cell, and then the cell wall, which is firm. But this plasma membrane is mobile and it's flexible. So these little blue dots are all phosphorus, and then in between here are the lipids. So it's a phosphobilipid layer. So it doesn't allow things in and out. So the plant can take and make proteins and put sensors that are proteins into that plasma membrane, which allows it to control what comes in and out. For example, here we go, there's that plasma membrane. Whenever you see those blue dots in that line, and it's permeable to ethanol, it's permeable to CO2, N2, and O2, a little bit to water, but not to glucose, not to cations, not to amino acids. So it doesn't allow them in unless it wants them in. And then when it needs something, say in the root cell, it actually has the ability to produce different proteins. These are called HATs, high affinity transport systems, and low affinity transport systems. So they both take a little bit of ATP, but notice this, under high affinity means that the whatever nutrient they're after, it takes a whole lot more ATP to do it. So the scarcer the nutrient or the scarcity of the product, the more energy the plant has to use under high affinity transport systems to get it. On the other hand, let's say a root tip is running into a sideband. Well, there's a ton of stuff there. This is just using potassium, but there's a ton of stuff there. So it doesn't have to use much energy. It uses just a tiny bit of energy to get that in, and it always pumps out some hydrogen in order to keep the cells on the exterior positively charged and on the inside negatively charged. It also controls water in that plasma membrane, controlling what goes in and out of water. So here we are back to that darn cell. And this vacuole is key. And I just point out here, the chloroplast and the mitochondria are the key ingredients. And we can see that in the cell wall, we have the plasma membrane, this blue line. So how does the plant do that? Well, those sensors in the plasma membrane tell the plant, I need something. So let's figure out, let's say we need some phosphorus, because I've been doing a lot of stuff on phosphorus. So it goes to the nucleus, finds some DNA that says, I'm going to make a phosphatase to help me get it out. So it makes a copy of that with messenger RNA, takes it into the to the ribosomes here, runs a whole bunch, makes up 10,000 phosphatase enzymes, runs it here through the endoplasm and shoves it into that cell wall, and holy camole, that plant is now excreting a exudates into the rhizosphere and extracting those nutrients. And with phosphates, it's chelating them so it doesn't get tied up, and it brings it back in here, and it runs it into the chloroplast and into the mitochondria, and it can run it into the next cell here through these little openings between the cells called plasmodesmata. Now, these plasmodesmata are critical because you can have a systemic response because the plant can communicate with all the cells next to it, and you can have that a cascade of events take place. You can also have hormones in one cell move through the other plants, and now they have something called microRNA, which are short pieces of RNA that can move from cell to cell to cell to cause a reaction in the plant to whatever it is. So the plant has that capability, and it responds genetically, and it does it automatically. It's like us. We eat, we breathe, our heart pumps, we don't think about it. It just happens. And the same thing goes on here. And within the cell plasma membrane and the vacuole, there are transporters that allow you to get into the cytoplasm. There are transporters in the cell wall or in the vacuole. And when the vacuole is full and has to give it back, it transports the nutrients that are in here back into the cytoplasm, and it's used. And the vacuole shrinks and shrinks and shrinks and shrinks. And when there's no more left to give up, you get deficiency symptoms showing. So the trick is to measure the amount of nutrient in the plant before it gets to that, because once you get to deficiency, the plant starts scavenging nutrients from lower leaves and moving them up. So that's our hidden hunger, and that's basically what we're measuring when we do a tissue test, is how much is in the vacuole, how much is in the cell. And if the whole thing is showing low, you know that the vacuole is running out of nutrients and it better get some roots going. So in the plasma, in photosynthesis, we have some ATP produced here, which runs into the Calvin cycle. We take in CO2 and water, and we give out sugar. Water has to be broken down here in order to occur, and that provides oxygen. That oxygen can't get out of the cell. It's stuck there because the stomata are closed. It becomes toxic, and what is termed photorespiration takes place, and it becomes toxic to the plant. So we have here, the first thing that happens in photosynthesis is water is broken, H2O is given out with sunlight. We then have, we have the transfer, we get excited electrons here, and that, if they're not captured, become toxic to the plant. We move it to plastocyanin, which is central atom is copper. Copper, about 30 to 40% of all the copper in the plant is associated with photosynthesis and electron transport. Then we have ferredoxin here, which is iron. You see how much iron canola takes place and why it's such a great plant, and it can pick up copper. All plants use the same process. And that gives you a little bit of hydrogen, which goes on over the Calvin cycle, but then we get ATP synthase, which makes ATP in the plasma membrane where photosynthesis takes place. So all these micronutrients, because I'll be talking about copper and I'll be talking about iron, these is, to me, this is where the damage is taking place, but we always talk about a copper deficiency, you know, in the leaf tips on wheat. But that's because the plant is not photosynthesizing, and it's, I think it's now causing an effect in those cells at the tip of the leaf, because it's the tips of leaves that are furthest away from the source, and the first place that there's going to be damage in the plant. Because if it damages the leaf close to where it attaches to the plant, it can't move nutrients out. So when you see tip damage, you know there's a problem arising. This just shows manganese in breaking the water molecule. This is plastocyanin. You can see it's copper, nitrogen, sulfur. Sulfur is key in many, many enzyme reactions. Take a look at the iron and ferredoxin here. Iron, Fe, sulfur, methionine, or cysteine, sorry. Cysteine. You almost always have sulfur hooking iron ions together. So, enough details. You don't almost have to remember all of that. You just have to know that all of those nutrients have a function, even though we don't often talk about it. And many of them are, or most of them, or all of them are at the cellular level, which eventually give you some symptomology of deficiency when there isn't enough for it to perform its function. So let me look at time. Okay, so here we go into this. So what happens in drought? So we get all of these factors: salt, flooding, extreme temperatures affecting the plant that sends a stress signal transduction into the DNA, as I said, and you get an altered metabolism. You get some new stuff produced in order to deal with, and you get a physiological change. It can be in the roots and it can be in the upper leaves all over the plant. The other thing it can do is it can change its root angle if it needs nutrients. It can form association with arbuscular mycorrhizae to help it with transport of nutrients to the plant. It can secrete organic acids into the soil. It can secrete phosphatase. It can secrete all kinds of enzymes into the soil, which extract nutrients and also feed the microbes that are mineralizing the organic contents around the rhizosphere. And that may be the critical part of the exudates, the carbons and sugars that are given by the leaf down into the root to form these acids, which are pumped into the rhizosphere to extract nutrients, but also feed the microorganisms those sugars that they need along with some minerals. It can make more lateral roots. It can grow more or longer root hairs. So it can put on advantageous roots, and that is controlled by a whole bunch of hormones that just come into play automatically, providing we have the minerals there to allow the plant to synthesize those. So what causes the stress in the end? Biological, well, we know diseases and so on take place in there, but the oxidative stress, and I got to read this, reflects an imbalance between production of reactive oxygen species, ROS. If any of you want to follow this up, just type in reactive oxygen species in Google and you'll get a pile. And a biological system's ability to readily detoxify the reactive intermediates or to repair the resulting damage. That's what we're going to talk about now. So we get all these biotic things, and we get all of these abiotic: flooding, heavy metal, intensive light, ozone, drought. These are all water deficiency, herbicides. All of that causes abiotic stress. And this is targeting at farmers, plus abiotic stress factors. For example, the human being, the farmer, is compacting the soils, not dealing with deficiencies, putting on excess fertilizers in rows sometimes, putting on pesticides that cause stress on the plant. You know, those big white blobs that occur on leaves when herbicides are put down or burning, those are stressful. Poor seed quality because the nutrient content of the seed is poor. Poor drainage. When you don't get drainage, the roots are in excess water and they can't get oxygen. Salinity, acidity. I was amazed. Last week in Saskatchewan, I've been hearing for almost ever since I got into even thinking about nutrients, that Saskatchewan was largely calciferous and high pH. Well, I find out there's some exceptionally low pH, under 5 in Western Saskatchewan that are needing lime real badly. And then the rotations, we're pushing the rotation. So between all those abiotic things and biotic things, we have the human being, which I guess would be a biotic thing, that are putting stress on the plant. We got pHs, and they influence what's being taken up. And here we got pH variance and like high pH through most of Saskatchewan, but you see this hot spot here? Like this is really hot. These are really hot in terms of acid soils. All of northern Alberta almost is acidic. Doug Penny's about 6 million acres, I think, in Alberta alone that is acidic, and we need lime on those. So all of this is affecting us. Soil phosphorus is low. Without that, plants are under stress in terms of, especially when it's dry and it can't get that phosphorus extracted. Zinc is low, and zinc is really key in response to stress, and I'll get to that. So these are zinc tissues are low. We're getting nutrients stratified all the time. So the plants, when it gets dry or they're poor roots, it can't get down to get nutrients. It varies across the field. Look at these levels here, sulfur, sulfate. And this is Ross Mackenzie's work where he just went across the field and took samples across the field, looking at variance. Nitrates weren't exceptionally bad. Look at the phosphorus difference, the potassium difference, the sulfate differences, the pH difference, almost 2 points within the same field. So we got plants that are under stress in one part, doing just honky-dory in the other part, and that's where you get the difference between your landscape and the kind of response you get on that yield monitor when you go across the field. And that's where we're getting into precision agriculture to try and nullify that. Too much urea. This is one of my favorite shots that I took this year. This is a crystal green granule with canola roots chewing it up. So you want that, you don't want this. Disease organisms are always there chewing away. This is stripe rust chewing away at your leaves, also causing stress. And the stress response always costs the plant, always is costing the plant. So when we saw those, the effect of abiotic stress and why it's so damaging is because it has to deal with all of that, and it all takes energy. It takes away from producing seed and big seed and lots of it. This is stressful on the plant when it's fixing or making the nodule, because the plant makes the nodule, so it actually takes a lot of juice. The relationship with mycorrhizae takes energy. So how do the plants respond? So what we have is all these stresses producing, these are the reactive oxygen species, hydrogen peroxide, superoxides, and then we get oxidative stress, and that causes molecular damage and a cellular effect, and the plant deals with enzymatic antioxidants and non-enzymatic antioxidants. You've probably all heard about it. With us as human beings, we have reactive oxygen species. We call them free radicals. Basically, humans use the same approach that plants do, and we get most of the nutrients, or almost all of the nutrients, from the plants who use the same process. So they take them up, or we get it from the animals that eat the plants. This is a little bit more complex. I'll go through about 3 different approaches to this. So we get this formation of all these reactive oxygen species right here, okay, and the antioxidants scavenge them. So we have enzymatic antioxidants, superoxide desmutase, catalases, a whole bunch of other things here I'll get to, and then we have non-enzymatic antioxidants, of which the biggest one is ascorbic acid, glutathione, tocopherols, which is vitamin E, and then a whole bunch of flavonoids and alkaloids. Another way to look at it is this. We have the enzymatic antioxidants, superoxide desmutase, catalyzed ascorbic peroxidases, which is fine. Those are all enzymes. They end in AS. Then we have the natural ones, the carotenoids. Proline is an amino acid that builds when the plant is under cold stress. Vitamin E, vitamin C, glutathione, which is coming from sulfur. So they have 2 approaches, and they work together. They work together. Some have abilities to take one of the superoxides. Some have the ability to take other ones, and most of them try to convert it to hydrogen peroxide. And then catalase can break that down to water and oxygen. Okay, so we get these environmental stresses going on, and it depends on how long, the number of exposures and so on, and which organs are affected, the stage of development, and the genotype, and we get either resistance, or we get susceptibility, and we get cell death or plant death. Simple, just like in human beings. When we get these active oxygen radicals here, we get membrane damage, DNA damage, we get chlorophyll damage, and we get protein damage. So we get cell death. And that's great. Remember that disease on the leaf where we get stripe rust? That's great if we kill the cells around it. But when we close the stomata for a long period of time, when we stress it for a long period of time, these reactive oxygen species build up and build up and build up and then start killing the plant itself. Mutations, chlorosis, protein damage. That's the problem. Now, the reactive oxygen species signal the hormones, and the hormones help us, and they control some of these. You can see we can salicylic acid, hypersensitive responses. We get jasmanic acid, which produce lignins in terms of protecting plants. We get gibberellic acids formed, which help with seed germination. We get all of those things, and we get salicylic acid affecting osmotic stress. So all of these things under low concentration are doing fine. When we get lots of it, here again, I love this shot because this is called the ascorbate glutathione pathway, and it's the key responder to all of these things, and these occur mainly in the chloroplast, mitochondria, peroxisomes, and you don't know the apoplast is in between the cells, and these are little cells within little organelles within all systems where peroxides are handled in the plant. And again, we get those enzymatic scavenger superoxide desmutase, ascorbic peroxide, glutathione, so on. And then we get glutathione here, ascorbic acid. So it's very clear. We get non-enzymatic, we get enzymatic. And when the balance is right, we get a healthy plant. When we get lots of antioxidants, when it's not, we get a disease. And here's just an example of how antioxidant enzymes and tolerance in plants, how the research has galloped on all these SODs, catalases, ascorbic acid, glutathione. Like, we have seen the massive amount of research go on these because now it's recognized just how important dealing with that is. And you can see that here we get the antioxidant, antioxidant enzymes like superoxidase, superdesmutase, and I'll talk about, we get chilling, high CO2, high light. All of these things affect the production of this. So the plant responds with, for example, glutathione is upregulated, chilling, drought, radiation, heat stress, high CO2 levels, and so on. And here it just shows again which ones handle which reactive oxygen species and where they're located. You don't have to know that, they're sort of everywhere. And now what we're looking at here in a research perspective is to take genes that are upregulating, say, ascorbic peroxidase from Brassica campestris, which is, a Polish rape, and trying to move them into other plants. And we're getting superoxidase genes. So these enzymes are all regulated genetically in the plant based on the response to the stresses. Easy. So here we go, your plant nutrition. How do we handle this? We're going to make it just about on time in an hour. Adequate level of K and C nutrition enhance plant drought resistance, water relations, water use, plant growth. We know potassium is key. We know calcium is key. In addition, to K and S, various micronutrients, including zinc, copper, iron, manganese, and nickel, have also been shown to be involved in detoxifying oxygen radicals. So here's what the definition: superoxide desmutase, an enzyme that alternately catalyzes the desmutation or partitioning, breaks it out of this superoxide radical into oxygen or hydrogen peroxide. So here we go. You didn't have to remember that last slide. Superoxide desmutase, and there are 4 of them. Well, with nickel, there are 5. So it takes these free radicals, hooks them up with hydrogen, produces hydrogen peroxide here, and then hydrogen peroxide is dealt with by catalases, peroxidases, no problem. So here they are: manganese SOD in the mitochondria, iron SOD in the chloroplast, copper zinc SOD, which is the most important, most widespread because it's in the chloroplast, and the cytosol, or the liquid in the cell. And they always work together, copper and zinc. Now, it's the copper that actually does the work in reducing these electrons, or scavenging these electrons, and zinc is part of the bond. Here you can see it's copper and zinc. So when Jan talked about copper problems, and how the plants suffered and how yields were low. It wasn't always because it wasn't in the electron transport system, but without copper in the plant, it could not deal with stress. And that's, to me, what brought down those plants in the early days that didn't have the ability to pick up copper. And still today, copper is a big chunk, along with, of course, zinc and those other micronutrients in managing that stress. And here, this is on Wikipedia. You don't have to, go very far. This is copper, zinc, nickel, manganese, iron. These are the way they classify and show their molecules. So, a lot of times you'll hear that superoxide dismutase, SOD, is a very important enzyme that functions at the cellular antioxidant level. Zinc is required for detoxification of toxic radicals. Well, people that were pushing zinc said that, but it was actually the copper and the zinc that was key, and the copper was doing the reducing. This was just the support and the building blocks. So, which plants can deal with stress best? Well, barley has a small root, so it has trouble getting nutrients. Faba beans have pretty small roots, so under drought stress, faba beans don't do so good. Maize has got a pretty good root, big root. This is kilometers of root mass per meter squared. So there's lots of it. Look at old canola. This is old Polish stuff. We got roots. We got real roots. Soybeans. How well did your guys do in soybeans last year when we had drought? Not much of a root system there. Really tough to get started. Very poor root hairs as far as I'm concerned. And look at wheat. So wheat and canola, the two crops that make Western Canada, have huge root systems. Rape has got the best extractive power of anything. It sucks the ground out of zinc, copper, phosphorus, you name it. It has root exudates and acids that can extract from organic matter a whole pile of stuff, in spite of the fact that canola, the brassica family does not have an association with mycorrhizae. It has developed strategies that are really good at mining the soil. And so does winter wheat and wheat. So those plants with smaller root systems will probably benefit a whole lot more from things like mycorrhizae and support than things like canola. Sulfur. And you've probably noticed, because I pointed this out several times, sulfur doesn't show up on this because sulfur has really no role in terms of interfering with other nutrients as far as we can tell. It's used by a lot of things. And you can see that the sulfur level, this was back in 1990, was massive. We were getting acid rain, so we started cleaning up our stacks. So here's 2000, it was still pretty bad. Here's 210, 215, and we're now putting sulfur back on the land in Ontario on alfalfa, because now in the northern hemisphere, where we've cleaned up a lot of our sulfur, sulfur is short everywhere. So sulfur, what does it do in plants? Enhances root development, increases productivity, well, that's okay. Promotes biological nitrogen fixation, it's true. Increases uptake of NP and K. Works against different stresses. Increases photosynthesis, synthesizes amino acids because there are two of them that have sulfur. Activates different vitamins, helps in chlorophyll formation. And sulfur derivatives give us cysteine, methionine, glutathione, abiotic stress tolerance through ROS and MG detoxification, abiotic stress management, heavy metal detoxification. For you guys that have been putting down elemental sulfur, or you've been putting down ammonium sulfate, although that's got a high salt index, or you're putting down biosol, I think one of the biggest benefits you're getting is, of course, sulfur. And here's the problem with sulfur for us. Look at canola. Huge, huge sulfur uptake. Huge, huge sulfur compared to anything else. Look what we've done with acreage, 6 million to now 21 million. Is it surprising we have a sulfur deficiency problem? And you look at the soil organic matter, which is the major source of sulfur, the retainer of sulfur, it's pretty low and decreasing here, especially in this area. So sulfur is key to that whole process. And this just goes to show that, just to exemplify, because I'm always trying to teach plant physiology while I'm giving my talk on anything. And we'll just go through this. Here's sulfate. The plant can't handle sulfate. It has to reduce it. So it brings it in through the root, stores some in the vacuole, put some in the plastids here, which are fine, and then send the rest of it up into the plant, into the leaves, on the xylem. There are transporters that take it there. Some of that goes into the vacuole. Some of that goes into the chloroplasts, becomes glutathione. You've heard me talk about glutathione. Glutathione moves here, and glutathione moves down in the phloem. And the sulfur and cysteine go into the mitochondria. Well, it just so happens that we take a look at this again in the reduction to sulfide before it can be absorbed into the plant and utilized. And you can see some of it is stored in the vacuole. And here are the transporters that bring it into the plant. And here are the transporters in the vacuole that can release it from the vacuole back into the what we call the cytosol, or the liquid of the plant. The first product that's produced out of sulfur is cysteine. From cysteine, it manufactures methionine and it manufactures glutathione, one of the prime scavengers of reactive oxygen species. And that is what the plant circulates through the xylem and back up the down the phloem and back up the xylem to measure the level of sulfur in the plant and to respond to reactive oxygen species, along with vitamin C ascorbate is our first responder. That's the magic for me of sulfur. Here are the cysteine and methionine. You can see that there are the 2 amino acids with sulfur in them, the only 2, but are key for a whole lot of sulfur compounds. Basically, all sulfur compounds come out of cysteine and methionine in plants. And the glutathione, which we just talked about, signals sulfur status in the plant, what I said, pathogen response, heavy metal tolerance, antioxidant defenses, and resistance to foreign nutrients and chemicals like herbicides that may be toxic. Just covered all that. Show you this again. Active and reactive oxygen species, glutathione peroxide, glutathione reductase, ascorbate, and glutathione as well here in combination with ascorbic acid are the primary ways that the plant handles it, and so do humans. So do humans. I can tell you that is, if you look it up in any biology, you'll see it. We're down to the last one, potassium. But of all the mineral nutrients, potassium plays a particular critical role in plant growth and metabolism and contributes greatly to the survival of plants that are under various biotic and abiotic stresses. Now, I didn't write that. That comes right out of textbooks. We have huge potassium uptake in peas, more so than any of our other crops, basically. So that gives you a clue. If you're growing peas in a low potassium area, look at doing something. It doesn't move very far. It gets tied up very easily in the soil clay lattices, but it moves further than phosphate. Phosphate is even less, and it doesn't move as far as calcium or nitrate. So I know we're running into potassium deficiencies later in the season simply because it's getting dry. But potassium is key in terms of regulating of the stomata, the opening and closing. These are stomata in I should tell you now, you know, that dicots dicot is not a term anymore. It's historical. The new term for those plants that we used to call dicots is eudicots and monocots. So this is the stomata of a dicot, eudicot, and this is the stomata in a monocot. And these cells here, the subsidiary cells, store a lot of potassium, and when the sun comes up in the morning, it puts that potassium in there and opens up the cells. So here's what happens in the morning and why potassium is really important, because it regulates stomata opening and closing. And you can see this is stomatal opening and this is potassium levels, the green. So 7 o'clock in the morning, the potassium level starts rising. because and that opens the stomata, follows that, and then sugar responds after that. At night, the stomata are closed. That's why on those nice graphs that Elston shows of water dropping, you can see those steps. Stomata are closed. When it opens in the morning, then you can see that drop in terms of water uptake. And it's the potassium that's key to regulating that in the signal from abscisic acid in the root zone up to the stomata. Then as sundown comes on, now that we got sugar processing, the stomata open, and it'll stay open, the potassium levels decrease till dark, and then everything decreases until dark, and then in the morning it cycles through again. Now if it's cloudy, real cloudy, you may actually not produce enough sugar in that daytime to compensate for what's used in respiration. That's for darn sure. And lastly, these are all the stresses that the way the plant handles stress with potassium: improves ROS balance and antioxidants, stimulates photosynthesis, deals with respiration, so regulates stomatal conductance and improves photosynthesis. K is really, really important. And you noticed in that one of the first slides that the amount of K going off in the seed isn't huge, but it's the number 2 amount in the plant after nitrogen. It stays in the plant because it's not actually incorporated into molecules. It's used to activate molecules, to provide turgor pressure to the cells and keep them hydrated, to regulate the flow of water in and out. And that is all key to stress management. So what can you do? You're farming for your children, so spend some money on getting rid of those acid problems and maybe looking at how you can deal with some of your real big issues. Again, tackle the big fish. Soil and tissue test, make decisions on data. Many of the micronutrients, I believe, should be going on a lot earlier. Things like boron should be going on earlier in the season. Things like copper should be going on earlier in the season, because those are important both for cell turgor pressure and this whole reactive oxygen species issue. Look after your soil. Get rid of those acids. Get rid of compaction or deal with compaction. Don't let it get worse. Don't let your plants run out of nutrients, especially the macronutrients. You have foliar sprays now that you can apply at any time and get a certain amount in. And I look at foliar sprays as a trick mechanism to grow more roots. Because if the plant doesn't grow more roots within a week, on a micronutrient application, it's back in the same boat. Growing more roots puts out more exudates into the root zone, feeds more microbes, mineralizes nutrients, releases them for uptake in the soil. Look after your rotations. If you go canola on canola, your mycorrhizal population has been darn near depleted. So try and keep that rotation going. Intercropping is a big thing, and that's a whole issue on itself. We probably need a webinar on just intercropping, but the idea is to keep different roots there so that you get exudates that are acidifying, basifying, and so on, and can control what's going on in the soil. Seeding, seed, dressing, and disease, look after those. Look after these nutrients is key. All of them are key. And think like a plant. Thanks a lot.